I have the absolute pleasure at this time to introduce Ambassador Deborah Birx. Dr. Birx has dedicated her life to improving the health of others around the world through the development of diagnostics, therapeutics, and vaccines in critical use to prevent disease and promote global health security. She was a past White House coronavirus coordinator and oversaw implementation of the U.S. President's Emergency Plan for AIDS Relief, also known as PEPFAR. Dr. Birx will be presenting Bacteriophages, a new and old treatment for antibiotic-resistant bacteria. Dr. Birx, the floor is yours.
Thank you. I'm just so impressed how many people have tuned in on a Labor Day weekend on a Friday afternoon. It's about 11:15 here. I'm in Israel for a wedding, but when Kevin asks you to do something, you don't tell him no. So hopefully we'll all stay awake, including myself. So thank you, Kevin. Thank you, Jeannie, for your excellent introduction. And what a great, I heard the measles talk and I heard the most recent talk. I'm just so thrilled that people are understanding the attack rate in measles. I know you all watched what happened in Gaines County. That was ground zero of the Texas outbreak. What the story wasn't told is all the counties surrounding Gaines County had a 1,000th risk of transmission compared to Gaines County, which was about 20 per 100. All the other counties really held, and they had vaccination rates around 95%. So we never tell the good side of the story, and really grateful to the parents out there that ensured people got vaccinated. And this airborne issue and the understanding that just walking in a hallway in a hospital brings bacteria, viruses, and fungi up into the air that then circulates. It's much more complicated than we ever thought, and I just thought that was such an excellent talk. Okay, we're going to talk a bit about bacteriophages. I'll go quickly. So we all know that there's increasing antimicrobial resistance. Bacteriophages have existed in nature forever, and they constantly adapt to their bacteria. Like all viruses, they're very receptor driven, so they can only infect their specific bacteria. So a Pseudomonas aeruginosa phage can only affect Pseudomonas. A Staph aureus phage can only infect Staph aureus, can't infect Staph epididymis. And what's really exciting is it protects your gut microbiome. And for the first time, I think we're beginning to understand how important your gut microbiome is. So the big issue that has plagued bacteriophages is, and this is a billboard that someone sent me from Georgia showing, this is an advertisement from Georgia and in the Ukraine showing that they've been using bacteriophage for a very long time, but didn't build the evidence base through a classical placebo-controlled trial. So we don't have any bacteriophages approved into the United States. So there's a sense that these could be incredibly effective, but they're what we call biologics, which means they have to be alive. So you have to grow these viruses in their host cells. What are their host cells? Well, I told you they're very species specific. So if you want a Staph aureus bacteriophage, you've got to grow it in Staph aureus. Now, everybody on this call knows that bacterial cell wall protein causes huge reactions if you inject it into the intravascular space. And so you not only have to produce the bacteriophages in a sterile way, but you have to ultra purify them away from what they were grown in without losing any of their potency. And this is what has taken the field so long. There's been great papers out now on how to develop phages, how to manufacture phages, and how to ensure, because they're a biologic, that the phages are stable. The work that people did for CAR T cells for cancer therapy, which is also a biologic, has really helped the phage field because it's really helped the FDA develop standards on what a biologic needs to look like to utilize. And so just going to talk briefly about each of these issues and finish with the phase 3 trials and how you can be misled, just so you can kind of see some of the data during these product developments. So as I described, you figure out how to grow this phage and optimize it, and then you have to move it from the bench into a GMP sterile environment where you can produce it. And so how the phages work is they bind to the host cell, they're bacteria species, they actually, like all viruses, can't reproduce outside of the cell. So can't reproduce outside of that bacteria. So they have to inject their genetic material into that bacteria, and then they replicate in that host. They can replicate at incredible speed. So they can replicate and produce all of those phages inside that bacteria and lyse that bacteria in about a 20 to 25-minute life cycle. So it's one of the few things that actually can keep up with the replication rate of bacteria in the body. And so when you manufacture these, you've got to infect the bacteria with just the right concentration, and you have to what we call ferment it till you get maximum lysis of the bacteria in that fermentation vessel so that you have maximum number of healthy alive phages. If it goes too long or it goes too little, the phages are either stuck inside the cell or the media that you grow it in gets less favorable for expansion of the phages. So you can see we know a lot about bacteria and viruses now, and through this whole process, you got to keep your staph host cell from mutating, and you have to keep your phages from mutating. And that is the critical elements to, because any changes in your phages, any viral mutations could change how those phages work in the human body. And you want reproducibility for the FDA. So just very quickly, these phages are grown in their host bacteria. There is a crude lysate from that fermentation, and you've got to take that very dirty lysate shown by these multiple other dots plus the phages, and you can see the tail fibers are thought to be the receptor areas, and the capsid is where the genetic material is. And you've got to clean up all the contaminants and maintain a structurally competent functional phage. And so that's the work that many companies have been doing over the last decade. But you have to prove the phages work. Now, I just got to cover clinical trial design very briefly. There's what we call superiority trials. That's saying your drug is better than all the other drugs on the market. Then there's a non-inferiority trial. So if you wonder, why do I have 10 antibiotics for Staph aureus? It's because there's been a series of non-inferiority trials where you compare it to another antibiotic and say it works the same. So it becomes another antibiotic on the shelf. Because phages have been difficult to make and difficult to purify, there's been a lot of compassionate use, because if you're dying from an infectious disease, people will accept higher side effects. But you can't really use compassionate use data to convince the FDA to have approval. And so the field has been doing compassionate use in Europe and in the United States, but it's never going to be a pathway for approval by the FDA. In compassionate use, purity and reproducibility is not as critical because you're making it for that very sick patient in that one moment. These studies have been pulled together. This is a study out of Europe where they studied 100 consecutive compassionate use cases, just to give you a flavor for it. And they did see that with antibiotics, the patients that received phages plus antibiotics did better than those that just received antibiotics. Now, superiority trials. There was a recent superiority trial called exacerbase. The name of it is not important. It was a lysine that thought that it would help lyse Staph aureus bacteria, and along with antibiotics, these patients would do better because Staph aureus obviously has a high mortality and very high morbidity, and often patients are in the hospital for 3 to 4 weeks on high dose broad spectrum antibiotics. So in this trial, they enrolled people with Staph aureus bacteremia, and many of them had endocarditis. A lot of them had MRSA, in other words, were methicillin resistance, and they studied the clinical response. And I'm going to show you the phase 2 data so you can see why they did the phase 3 study. So they looked at the response rate at day 14 at the end of the trial and then 28 days after the end of the trial to evaluate whether these patients relapse. Because patients with complicated bacteremia have an extraordinary high relapse rate of between 20 and 25%, and sometimes up to 30%. And so most of these patients were enrolled in the United States. They enrolled 116 evaluable patients. This is just their demographics. A lot of the antibiotic treatment was vancomycin or daptomycin for MRSA, and third and first generation cephalosporins and penicillins for those that were sensitive. And just this quick slide to show you the demographics were matched between the two. And what they found, although they didn't find an impact in the overall treated population, in the subgroup that had Staph aureus bacteremia with right-sided endocarditis and those with MRSA, the second line from the bottom, did much better. So they launched a phase 3 trial utilizing very similar endpoints. And the bottom line of the whole story is when they did the study, they didn't find any advantage to having exacerbase plus antibiotics versus antibiotics alone. The response rate at day 14 was 60% in the standard of care and 50% in those that received the antibiotics plus exacerbase. Why am I explaining this all to you? Because there has not been a successful superiority trial in infectious disease for a new product in 2 decades. So we've been doing a lot of non-inferiority trials, adding new antibiotics for which Staph aureus is becoming resistant, but we haven't done a superiority trial to show that a new agent could actually work. I just wanted to highlight something to you all, and I'm sure you see this. Clinical trials can have significant biases because of patients' willingness to enroll. And so this is just a cancer study. And what happened was the patients that had lower grade lesions and lower grade stages were more likely to enroll than the sicker patients. And so you can end up not having an effect or overreading the infect because the patient population really doesn't match the spectrum of disease. And so clinical trials, and I'm just, for all of you that read them, really look to see, and you know clinically how these patients look in the hospital. Really ask yourself, is that representative of the patients that I normally see? Because it can lead to these biases. Now, these non-inferiority trials that I just described, a lot of the antibiotics, the new antibiotics are done that way. This most recent one, this ceftobiprole, that was a non-inferiority study against daptomycin. Now, this is the overall treatment success. This is after full dose of antibiotics in Staph aureus, only a 70% response rate. So when I'm talking to you about high levels of treatment failure in Staph aureus, this is very common. So you can see they had treatment failure rates of 30%. So this is why there's a call, a real push to not only get a new system or a new approach, but to really get to a place where we can actually cure complicated Staph aureus bacteremia. And this is the data that what you have to show in a non-inferiority trial is it's not worse than this approved therapy, which was daptomycin. And you can see they, from the beginning, set that confidence interval to say it has to be less than 15% difference. And so that's how they pick all of that. And that's how those studies are done. There are quicker studies. There are lower ends often. And so that's how most of our antibiotics today are approved by this non-inferiority trial. Now, you always have to look at what day they looked at the outcome because complicated bacteremias like Staph aureus can relapse. So although they had a high 7-day response rate, the test of cure and persistent bacteremia often was seen in these patients. So you have to kind of look at every single study to see which ones have the most impact of preventing relapse. There's an interesting study going on in Europe right now because people who treat patients with bacteremia, particularly Staph aureus bacteremia, it's just become standard of care that we treat for 4 to 6 weeks, but there was never a study to actually look at 4 weeks versus 6 weeks. So they're actually doing that study right now across Europe to really say, do these patients need 4 weeks of broad spectrum antibiotics or do they need 6 weeks? And that's going to be a very important study because less high broad spectrum antibiotics will lead to lower C. difficile and complications from having all of your normal flora depleted. And so their primary outcomes is a patient alive and then no evidence of disease. So we have been working on these bacteriophages for these complicated Staph aureus bacteremia to show that phages are with antibiotics initially are better than antibiotics alone. And so those are the trials that we've been doing. I won't go through everything that the company does, but we've had really good support primarily from the Department of Defense. We've looked at Pseudomonas aeruginosa as an inhaled bacteriophage cocktail, and we've looked at Staph aureus bacteremia as an IV injection every 6 hours for 5 days. I'll just quickly talk about the Pseudomonas program. The big issue with Pseudomonas and non-CF bronchiectasis, both CF and non-CF bronchiectasis have big structural abnormalities in their lungs, and they end up getting opportunistic infections like Pseudomonas that becomes a chronic infection and leads to multiple exacerbations. We did find a statistically different level of Pseudomonas in the lungs for people who were treated for 10 days. It's statistically significant in the analysis for people who were treated with inhaled Pseudomonas phages. And that was seen in both what we call a cohort A against all the placebos. Those were individuals not on antibiotics. Cohort B was people who were on inhaled antibiotics to which we added phage. To me, the most exciting program right now that we're working on is against Staph aureus because it remains quite a deadly and high morbidity. I mean, just really difficult for these patients that are on broad spectrum antibiotics for this long. Most of them are over 65. A lot of them are on dialysis. Some of them have endocarditis. So it's a really often complicated and deadly disease. And so we looked at outcomes at day 12, at the end, 1 week after stopping antibiotics, and at the end of study, which was 4 weeks after stopping antibiotics, to look for relapse. Importantly, remember I described how you have to have this really pure or you end up with severe reactions. We did not have any severe reactions or what we call severe adverse events to the study drug. We did have a hypersensitivity, and that was thought to be related to the vancomycin. And we had a patient that had a few days of an elevated transaminitis that returned to normal. And so we didn't see, it's just our way of proving that our phages are really quite pure. So this is outlines the study and when we look for response. So here's the bottom line. We had a blinded PI at each site that assessed the patient clinically and all their signs and symptoms, and they had to have 100% improvement of all their signs and symptoms in order to be determined as a responder. This is the day 12 response, and you can see there is statistically significant improvement. The red bars are those that didn't respond, so we had a 58% response rate. If you remember back to the first set of slides that I presented in non-inferiority trials, that's kind of been the day 14 response rate. And then at the end of the trial, we had no relapse in any of the patients that received phage, including those that had endocarditis, both right and left-sided endocarditis, which I think is quite important. And so these relapse rates of 23 to 25% are the common relapse rates in other trials. It's always important when you're doing a clinical trial to show that it's consistent with the trials that have been done to date, because you want to be able to put we don't want to be misled by a phase 2 trial. Phase 3s are very expensive. So you want to make sure that your data is consistent with prior trials and you don't over or under interpret the data. We know that MRSA subjects are particularly susceptible to relapse. And again, you see the higher relapse rates. We saw no relapse after end of antibiotics or 4 weeks later. And you can see a much higher relapse rate in those with MRSA and antibiotics only. Now, C-reactive protein is a measure of general inflammation. Obviously, when you're septic or you have bacteremia, bacteria in your blood, it does cause increase in inflammatory proteins. It's thought that C-reactive proteins are consistent with more severe disease. And you can see that both the placebo arm, the antibiotic alone, and compared to the antibiotic plus phage, started at very similar C-reactive protein levels of over 200. But in the phage arm, they were back to normal as a mean in the ones that received phage plus antibiotics and did not achieve that same goal until after the end of antibiotics. Just showing that with phage, you get an earlier cure. So in parallel, these individuals spent less time in the hospital, less time in the ICU, and of course did not have relapse rates. And we did not see a lot of study drug reactions as I described. So these are treatment associated adverse events. So I think what we're trying to do is to bring an evidence base that phage can change the standard of care. To do that, you've got to do a phase 3 trial. We're designing that right now based on the same outcomes of clinical response, but we will do day 14 and then, of course, a week after antibiotics and then 4 weeks after stopping antibiotics to really look at ensuring that we don't develop relapse. And I'm just really proud of the team who did all of this. I'm just there as an organizer of everybody. And so we have incredible scientists doing incredible work. And I'm really grateful to the clinics. You know, bacteremic patients are really sick. And to enroll them in a phase 2 trial is really difficult. And we enrolled all of these patients, the 42 patients in the phase 2 component, in just 10 months. And it really shows we had great site investigators. And these are just pictures of our labs and our fill and finisher so that we can manufacture in the United States and have control of our supply chain. So I hope I left you with a couple of concepts. I think most importantly, phages are biologic and they hold great promise. They're live actors. They can't infect your cells. They can only infect their specialized bacteria species. But in order to be effective and to be able to use in human, you have to carefully select those phages against clinical isolates of Staph aureus, which we did, and we have a 95% host range. Then you have to carefully manufacture and purify them while maintaining their potency. So you got to make sure that when you inject them into a person, they're going to be live and function. And what was exciting to me, it didn't matter if these patients had cellulitis or septic arthritis or endocarditis or an empyema. Those phages that we did IV push home to the site of that staph, found them, and then attacked them, reproduced. And we found phage back in the intravascular space, showing that they actually found their target, which was really important to me. Trials need to be carefully designed, and we all need to understand clinical trials so that we can look at the data ourselves. And we may need, of course, to show that the double-blinded and placebo-controlled. So thank you. Thank you for the opportunity to speak with you. And Kevin, really thank you again for setting this up. I'm still stunned that you have this many people online on a Labor Day weekend.
Yeah.
I couldn't bow out because I was like, well, if these people are willing to show up on a Labor Day weekend, then I certainly can figure out how to get online from Israel. You know, we're a little ahead of time. So I just wanted, there were a couple of questions in the chat. And so we administer, and I think what's important for systemic infections is to be able to administer it intravenously, even if it's pneumonia. We had patients with Staph aureus pneumonia and empyemas, and they had a response. But you asked a very good question also about pneumonia. we're now re-looking at our Pseudomonas product to reformulate it for ventilation pneumonia. I think, those are also incredibly sick patients. So we're just really excited about the opportunity to potentially have something that will make our response, even if we're using antibiotics, to really get a more rapid response, get people out of the hospital quicker, and critically not to have relapse. And hopefully my goal is to get to a point where phages are an alternative to antibiotics, because then you've protected people's microbiome. And I think we're finally beginning to understand that it's linked to Parkinson's and all series of diseases. We've ignored our gut microbiomes too long. So I think that's another really exciting area of research. If you're watching what's happening in Alzheimer's, they just turned the Alzheimer's totally on its head, trying to understand these accumulation of these proteins that may actually be due to infections and reinfections and changes in your microbiome. So I think the next decade in medical research is just going to be incredibly exciting.
Yeah, I mean, the big question is, how do you operationalize it? I mean, what sort of system would you have to set up, since each phage will have to be customized for the microbe
Yeah.
that the person has?
And that's why we're not doing it that way. We're doing broad cocktails that cover 95% of all staff, so that I think, like CAR T cells, once you show that it is effective, people will figure out how to individualize it further. But I think to prove the concept and to get it out there to patients, I think doing a phase 3 trial to show that you actually do prevent relapse, even if it was just used in that subgroup, that represents 25 to 30% of all complicated bacteremia. And it is incredibly discouraging to relapse. So this would be huge for them. And then you just keep working backwards, show that they have better outcomes, they need less antibiotics, you would like to go to 2 weeks of antibiotics. I mean, you could just keep pushing the envelope. I know where I want to go, which is phage without antibiotics, but, got to do it in a stepwise manner. So.
Absolutely.
From what I understand, phage therapy has the advantage that as the bacteria tries to adapt to the phage, it can actually become now more susceptible to the antibiotics. So
Yes.
resistant bacteria can become susceptible.
Mhm.
And that's
And
one of
these
the advantages.
phages are very, now remember, they're killing bacteria every 22 minutes. And so their replication rate is so high that they're able, if staph tries to mutate, it can mutate right along with them. And so that's the exciting part. It's like in vivo amplification, mutagenesis, because it's spent a lifetime living, trying to figure out how to get inside those Staph aureus. So it has really figured it out. And so we're taking advantage of what nature has given us, but really in a scientific way.
Yeah, and I think, as illustrated in your talk, in Eastern Europe, this is standard care. They would be attending this webinar thinking, well, what are these people talking about? This isn't experimental. We've been using it for 100 years.
Yeah. Yeah. Well, I know, but I appreciate the FDA and their stringency. So if you want a breakthrough, you've got to do a superiority trial. And I think being able to do the first potentially successful superiority trial in infectious disease would be just an incredible capstone to all of my work with viruses. So I'm kind of excited about it.
Yeah, and I like phages because, we talk about bad viruses all the time. We need to realize there's actually good viruses. We actually need them. Viruses are part of nature, just like the gut microbiome. We can't sterilize everything. Bacteriophages are actually beneficial.
And really appreciate the audience and the speakers. Really great talk, other speakers. They were really fantastic. Thank you.